Louise?
Yeah.
It's your stage. But I've forgotten. The very nice drawing that you see on the front page is a drawing by Louise, and kind of a wonderful drawing. Okay, Louise, it's your part.
Thank you. Thank you for the very nice introduction. So I would first like to thank the organizers, so Axel and Christopher, for accepting my participation to this series of seminars, because it's a great honor to be present among those great researchers. So yeah, today I will talk about part of the results I obtained during my PhD, which was, as Axel said, about the effects of the short neuropeptide F on honeybee behaviors, and mostly behaviors that are related to motivation. So I will start with a very general introduction, saying that we all know that behaviors are flexible and subject to modulation by various factors such as the internal state, experience-dependent plasticity, or the external stimuli. And a key concept in the modulation of behavior is actually the motivation, which has been defined as the energizing of behavior in a pursuit of a goal. So the general idea is that motivated behavior leads to a goal that is rewarding per se. So in fact, it has been postulated that behavior precisely occurred to reduce biological need. And later, the definition of motivation comprised so a goal-directed and an arousal component. The regulation of motivated behavior is achieved by the coordinated actions of molecules, such as neuropeptides, hormones, or neurotransmitters. And all of these molecules would act within specific circuits that will integrate multiple sensory signals to ensure the coordinated action between the external and the internal milieu. And all of this ensures that the animal is displaying the appropriate response in a given situation. So, like I said, neuropeptides play a fundamental role in determining motivational states in animals. But what are they? So they are a short chain of amino acids, and they would bind to a specific receptor, J protein coupled receptor. And they are mainly neuromodulators, but they can also act as hormone or local co-transmitters. And they are known to regulate many behaviors linked to motivational states. So here I wrote a few of them. And today I will talk about those three behavioral contexts because they were actually the strong focuses of the team where I did my PhD. And so we had all the tools to address them. And one family of neuropeptide modulates all of the behaviors that I wrote before, and they are called invertebrates, NPY. And so they have been very well studied in mammals, in rats, and in mice, for instance. And for instance, under starved conditions, NPY signaling would promote food ingestion and food-seeking behaviors. But it also plays an anti-nociceptive role, and it also regulates fear and anxiety under stress conditions. And so this would render the animal more resilient to stressful stimuli, for instance. And there's a few studies only that also highlighted a role of NPY signaling in learning and memory with positive and negative effects. And in invertebrates, one homologue has been identified. It is called NPF. And there's also an analog to NPY called SNPF, for short, neuropeptide F. So it means that they share the same function, but they are actually evolutionary distant. And here I just represented a few invertebrate species where NPF and SNPF have been studied. But of course, most of the functional studies have been done on Drosophila melanogaster. And actually similar results to that of mammal were found, with NPF and SNPF having a facilitatory effect on food ingestion and food-seeking behavior under starved conditions. It also plays, actually NPF also plays an anti-nociceptive role in the fruit fly, and also modulates the aversive long-term memory and memory retrieval under starved conditions. However, the functional studies of those neuropeptides on social insects are scarce. In colonies, for instance, foragers are highly motivated to meet the colony needs, such as the food collection or the defense of the nest. And these behaviors are actually regulated by social cues. So, for instance, in honeybees, you have the foragers that would collect food for the colony stores rather than for themselves, although both factors might be related. And in addition, some pheromones would trigger foraging activities. You also have associative learning and memory in both appetitive and aversive contexts that are required for foraging activities. And under stress conditions, the bees would actually release alarm pheromones that would in turn trigger the collective defense of the hive. But what do we know about SNPF and NPF signaling in honeybee foragers? Well, we know that both genes, NPF and SNPF, have been identified in the honeybee, but actually there's a singularity in the honeybees that there's no NPF receptor identified in the genome. So that's why during my PhD, we focused our attention on SNPF rather than NPF. There's two populations of neurosecretory cells that have been identified. So here you have a picture of the honeybee forager brain. And you can see here in black the cells, the neurosecretory cells of NPF and here of SNPF. So here you have the mushroom body calyces. And so there's actually only 8 to 10 medial neurosecretory cells for NPF. And in a different subpopulation, there's only 4 to 6 pairs of neurosecretory cells for SNPF. We also know that both peptides are expressed in the midgut, but the localization of the receptor SNPFR is actually unknown. So from the work of Ament and collaborators in 2011, they suggest that it is also expressed in the brain and in the abdomen. SNPF could also play a role in the phenotypic behavior as SNPF levels vary between nectar and pollen foragers when they arrive at the feeder. And finally, we know also the receptor SNPF is upregulated in foragers' brain when they come from food-deprived colonies compared to foragers that come from well-fed and healthy colonies. So the main aims of my PhD stand in those 3 questions. Actually, the big goal was to explore the role of SNPF on several motivated behaviors. So we wondered whether it modulated appetitive responsiveness, but also aversive responsiveness, like in Drosophila. And we next wondered whether it could affect the different forms of learning and memory. So I'm going to introduce now the method that we used for all experiments. So I'm just going to say it once, and then it's the same for the rest of the results I will talk about. So basically, we caught each forager individually in the apiary, and we put them in syringes like in this drawing. And then we brought them back to the lab and we separated the bees into 2 populations, to 2 groups. So one group was fed, partially fed, and the other was kept deprived of food. And then fed bees were either left untouched or they received what we call a topical application of a mixture with the solvent and the neuropeptide. So this is a non-invasive technique that consists of dropping 1 microliter of this mixture, so the peptide and the solvent, on the thorax of the bee when it's asleep. And then the solvent enables actually the neuropeptide to penetrate through the chitin of the insect and to diffuse in the bee's body via the hemolymph. So we had 2 groups of bees that received the topical application with the neuropeptide and the solvent. We also had a control group just treated with the solvent, and then the starved bees were left untouched. So this protocol is actually the results of months of adjustments made by my former supervisor, Gabriela de Brito Sanchez. So this protocol was ready when I started my PhD. And so before presenting the first results of the PhD, I would like to talk briefly about an experiment that has been recently done by David Baracchi and Gabriela de Brito Sanchez at the University of Florence in Italy. So the experiment was done because when we submitted the papers of my PhD, the reviewers were asking for somehow a proof that the topical application was working. And we then decided to conduct this experiment. So the idea was actually to show that the topical application was delivering the neuropeptide and that SNPF was actually present in the honeybee hemolymph. So because it was conducted in December, we only had access to winter bees. But actually, Gabriela showed a few years ago that winter bees are behaving the same as what we call partially fed bees because they have high level of lipid storage. And so here we consider that our winter bees are equivalent in terms of ingestion or food, in food ingestion to our forager that have been partially fed. So they took for winter bees outside of the hive, and then they were divided into 3 groups, so that didn't need to be fed. So one group was treated with the solvent only, and the other group received the topical application with the neuropeptide. Then 30 minutes later, the hemolymph was collected and then prepared to be injected into the mass spectrometer. Really familiar with mass spectrometry analysis, but so from what I understood, you have here on this iron chromatograms the SNPF signal for at the lower level the fed bees that was treated with the solvent only. I mean, the winter bee treated to delete this, and on the lower part is the bee that received the highest dose of SNPF. And so you see here the red peak that corresponds actually to SNPF. So, and the other peaks here are actually just noise background. So we have this peak here when the bee is treated with the neuropeptide that is absent in the bee that is only treated with the solvent. So we can now say with confidence that SNPF is actually present in the hemolymph of the bee after the topical application. Okay, I come back to my different treatments, and now I'm going to present the experiments we did. So we first started with a very simple ingestion experiment. So we placed back the bees individually in syringes, and we gave them the possibility to drink up to 100 microliters of sucrose, 20% that was left pure, I would say, or that was spiked with salicin, 100 millimolar. So salicin, it's a bitter compound that is not liked by the bees when it's actually mixed with a sucrose or sugar solution. And so 1 hour later, we come back and we measure what's left in the tip of the pipette that we inserted in the syringe. So on the y-axis, you have the ingestion per bee in microliter. And you can see in red, the fed bees ate less than the starved bees. So this is what we expected. And the fed bees and the fed bees treated with the solvent behaved the same. They ate the same amount of food. So those are the results actually only for the sucrose, 20%. And fed bees, now that are treated with the lowest dose of SNPF, ingested more food than the fed bees. But when we treated the fed bees with the highest dose of SNPF, they did not significantly increase their food consumption. So now for the results with the sucrose that was spiked with the salicin, we see the same profile actually with the fed bees eating bees and the fed bees solvent behaving the same as the fed bees. But you can see that overall the bees ate less than in the previous graph. So it means that the sucrose spiked with the salicin is less appetitive for the bees than the pure sucrose solution. And when we did the topical application of SNPF, here the highest dose increased the food consumption of fed bees. So we can say that overall, SNPF increased food intake for both the pure sucrose solutions, but also for the sucrose that was less appetitive for the bees. Then we studied the gustatory responsiveness of bees. So each bee was stimulated with a succession of sucrose solutions of increasing concentrations, and they were spiked or not with the salicin, like in the previous experiments. So it's actually quite simple. You have the bee that is tethered, and with a toothpick that was soaked in the solution, you touch the antennae, and if the bee likes the solution, it will extend the proboscis. And then we calculate a score per bee. So for instance, if the bee elicited 2 PER for the two highest concentration, sucrose concentrations, then it has a score of 2. So you can here easily understand that the higher the score is, the more sensitive the bee is towards low sucrose concentrations. So for the results, here only for the sucrose, so you have the score of the bee on the y-axis. You can see that the starved bees had a higher score than the fed bees and the fed control bees. So it means that our starved bees started to respond to low sucrose concentrations, whereas the fed bees and the fed solvent bees only responded to the highest dose, the highest concentrations. And now when we applied our treatment with the neuropeptide, we saw that our fed bees increased their sucrose responsiveness. And for the results with the sucrose solutions that were spiked with the salicin, we see the same profile with the fed bees having a lower sucrose score than the starved bees. And we see here that when we did the topical application on fed bees, we did not reach the level of starved bees. We have a result that is somehow in between the fed bees and the starved bees. So something happens here, but it's not strong enough to reach the statistical significance. So we can say here that if we want to be a bit strict, that SNPF topical application only increased significantly the score of bees only for pure sucrose solutions. Then we tested the olfactory responsiveness. So again, we have our bees that are tethered, and they were placed in front of an olfactometer. And we chose to work with those two odorants, so linanol and 2-phenylethanol, that are known actually to trigger spontaneous PER response without any training. And so we tested every each bee with both odorants, and then we pulled the data because the response for both odorants were the same. So we can see here that, so you have the percentage of PER on the y-axis, and you can see that the starved bee responded very strongly to those two odorants, contrarily to the fed bees and the fed control bees. And when we treated the bees with the lowest dose of SNPF, we actually also increased their olfactory responsiveness, but not to the extent reached by the starved bees. So it suggests that this neuropeptide here does not act alone in the modulation of olfactory responsiveness. And we next wondered whether this behavioral change that we observed could also be translated to the neural activity of the primary olfactory center in the honeybee brain, which is the antennal lobe. So to help me answer this question, I requested the support of Marco Paoli, who's an expert in calcium imaging in insects, and he's actually a postdoc in the research of animal cognition in Toulouse. So before explaining the experiment, I will briefly remind you how an olfactory information is processed within the honeybee brain. So you have first odorant molecules that will bind to olfactory receptors that are located on sensilla that actually cover the antennae. And in these olfactory sensilla, you have olfactory sensory neurons that will convey the olfactory information from the periphery, so from the antennae, to the antennal lobe, which is the first primary olfactory center in the honeybee brain. And the antennal lobe is actually constituted of glomeruli that are the first synaptic interaction between olfactory sensory neurons and other neurons. Then the olfactory message is processed within the local interneurons that connect the glomeruli together. And then it goes to the lateral horn and the mushroom bodies via projection neurons. So the idea of the next experiment was to measure the activity of the projection neurons upon SNPF treatment. And so we used 2 phantom microscopes to answer this question. And we tested 3 odorants. So we used, again, linanol and 2-phenylethanol that are appetitive odorants, but we also added a 3rd neutral odorant, which was the nonanol. So first, we had our starved bees that were prepared for imaging, and then they were placed under the microscope, and the projection neuron activity was recorded for the 3 odorants. Then the bee was fed, and then it was either treated with the solvent only, or it was treated with the solvent plus the highest dose of SNPF. And then again, the projection neuron activity was recorded for the 3 odorants. So on those graphs, you have the glomerular response of the antennal lobe for the starved state in blue and the fed state for the 3 odorants, linanol, 2-phenylethanol, and nonanol. And so here you only have those 3 graphs represent the glomerular response when the bees were starved and then treated with the solvent only. So you can see that the 2 curves don't overlap. It means that the glomerular activity was, it's actually the projection neurons activity was higher when the bee was starved. And then when we fed the bee and treated it with the solvent, this activity decreased, as you can see here with the curve in orange. And so this is the same pattern for the 3 odorants, no matter if it's an appetitive one or a neutral odorant. Then here you have the 3 graphs for the bees that were starved and then treated with the neuropeptide. And the difference here that you can see compared to the graphs at the lower level is that the curves are overlapping. So it means that the treatment with the neuropeptide rescued the projection neurons activity to the level of the starved state. So it means that when the bee is starving, the projection neurons activity is high, then it decreased when it's fed. But then when you add the neuropeptide, when you increase SNPF level inside in the bee's body, then the projection neurons activity is rescued to the level of starved bees. So to sum up a bit the results for now, we showed that with this very simple and non-invasive technique, we increased the food ingestion of bees, but also their sucrose responsiveness. We had mitigated results when it comes to sucrose responsiveness spiked with the salicin. We increased the olfactory responsiveness and the antennal lobe odor response. So we wondered now if, like the Drosophila, SNPF signaling could also modulate aversive responsiveness. So to that end, we used 2 standardized protocols which elicited the sting extension reflex of the bees. So the first one was a series of increasing voltages, and the second was a series of increasing temperatures. And as for the gustatory responsiveness, we calculated the score per bee using actually the same method. And so the analysis of the individual responsiveness score revealed that there's no effect of SNPF on electric shock responsiveness. And it was actually the same for the thermal shock responsiveness. So here, contrary to what's been observed in Drosophila or in mammals, SNPF in this context and with these individual responses does not modulate the bee's behavior. So we can conclude here that in our experiments, SNPF did not modulate the aversive responsiveness. But if we come back to our result on sucrose responsiveness that was increased by the SNPF, it's quite interesting because when we look at the literature, we know that sucrose sensitivity is actually a determinant of cognitive abilities. So it means that usually when a bee has a high sucrose responsiveness, it correlates with a good learning score. So the bee is a good learner. And on the contrary, if the bee has a low sucrose responsiveness score, then it performs badly in cognitive tasks. So we wondered whether we could translate this effect on the sucrose responsiveness to the level of appetitive learning and memory. And we reasoned that it could have an effect on actually appetitive visual learning and memory formation. So I chose to work on this task because I wanted to go closer to natural conditions. So we used a small Y maze that have been designed actually by Alexis Buatois, who was a former PhD student of Martin. And in this Y maze, the advantage is that each extremity of the arm is detachable. So for instance, when the bee makes a choice to go towards one of the two colors that is presented at the extremity of each arm, then the experimentator can block the bee and then replace it at the entrance of the Y maze. So it's like a closed loop. So we needed this because we couldn't let the bee go back to the hive and unload the crop as it's usually done in real free-flowing experiments, because we needed to control the crop load, and we also needed to give the bee the treatment. So we had to choose, we had to make a compromise. So it's like a real free-flowing experiment, but within a closed loop. And so we chose to work with those two colors, the yellow and the blue. And here, as you can see on this graph, you have the chromatic contrasts with the gray background, because each color was stuck on a gray background. And so you have the gray background in the middle, and the further away the color is from the background, the more the higher the contrast is between the gray background and the color. So you can see here that our yellow contrasts more with the gray background than does our blue. So we started already with this kind of unbalanced saliency between the two colors. So this was the protocol. So we had first a familiarization phase where actually the bees were free to enter the Y maze and to explore. We had bees that were walking and flying within the Y maze. And then once a bee had explored both arms and take the sucrose solutions that was placed at the extremity, then the bee was removed from this training Y maze and then placed in the real Y maze. I mean, it's actually the same, but in another Y maze to perform the conditioning. So before starting the conditioning, actually the bee was fed and then received the treatment, or it was left untouched if it was a starved bee, for instance. And then we had 10 conditioning trials, followed by 1 hour of rest. Then we had 1 memory test, then 2 refreshment trials, and then a second memory test. And so I would like to precise that those experiments were actually done by an intern, Emmanuel Briard, and also by Martin, who was willing to help me finish this experiment. So we first analyzed the learning curves, and as you can see, overall, we did not see a clear effect of the nutritional state, nor of the treatments with the SNPF. So on the y-axis, you have the percentage of correct choice, meaning, so we had bees that were trained to sucrose reward with the yellow color, for instance, and other that were trained to associate the blue color with the sucrose. And so here you have both groups, the yellow, the one that were trained on yellow and the one that were trained on the blue, that were plotted together, and you can see the percentage of correct choice. And overall, the only significant results we had was here, where the pea-fed bees treated with the highest dose of SNPF had a significantly percentage of correct choice that was higher than the level of chance, which was 50%. And next, we analyzed the memory test. So you can see here in the middle, the starved bees that were actually quite good because they remembered for both tests that the yellow was associated with the sucrose. So it means that they chose during both tests to go towards the yellow. And it was not the case of the fed bees and the fed bees treated with the solvents. And for our bees that were fed and then so less motivated to search for food, the one that received the highest dose of SNPF actually performed as good as the starved bees because they also remembered that the yellow color was associated with the sucrose reward. But it was not the case for the fed bees treated with the lowest dose of SNPF. However, when we conditioned the bees with the blue color, nor the starved bees, nor the fed bees or fed bee solvent expressed memory. So it was, when we look at the chromatic contrast, it is not very surprising because, like I said before, the yellow appears more salient to the bee compared to the blue. And so what we see here is that the starved bees did not overcome this saliency, although the blue was rewarded with a sucrose reward. And in those results now, we can see that the fed bees treated with the highest dose of SNPF did remember the association between blue and the sucrose for the second memory test. So here we can only conclude that SNPF is acting in circuitry that are potentially involved in the memory formation and retrieval because it improved the visual memory of bees. So to summarize, we can say that our neuropeptide also increased visual memory acquisition and formation. So with all the results I've presented so far, we can say that we have now a rich and multifaceted view of SNPF on the behavior of a social insect, which was lacking before. But we have not really discussed about the mechanisms that are involved in the SNPF signaling. So for the next few slides, I will discuss what kind of other signals can act with SNPF to actually modulate all the behavior I've presented so far. So I will briefly discuss the neuromechanisms that are involved in taste perception, olfactory perception, and visual memory formation. So we thought that SNPF signaling could act at the peripheral level for the taste detection. So it means at the level of the gustatory receptor neurons. We can speculate this because we saw that it increased the ingestion and it also increased the sucrose responsiveness. So we had a facilitatory effect overall. And but we have an asymmetry concerning the results with the sucrose when it was spiked with the salicin for the gustatory responsiveness. And those results were actually surprising if we compare those with the results obtained in Drosophila. So there is a very nice paper from Inagaki and collaborators in 2014 where it showed that starved flies would exhibit, would be more sensitive to sucrose and less sensitive to bitter compounds. And those modulations are actually regulated via independent neuromodulatory pathways. So for instance, when a fly is hungry, you have NPF that would act on dopaminergic signaling that would in turn increase the sensitivity of the gustatory receptor neuron that are tuned to to sucrose or sugar testing, and it would promote the food intake. In another pathway, you have SNPF that is acting on GABAergic signaling, and this signal inhibits the bitter receptors, the bitter gustatory receptor neurons, so it will promote the food ingestion as well. So with Gabriela and Martin, we thought about what could happen in the honeybee brain, and there's a few possibilities. So in a recent paper, Gabriela showed that the adipokinetic hormone that might kill the Drosophila, actually there's no effect on food intake and gustatory responsiveness in foragers, because adipokinetic hormone is mostly in the lipids, fat storage, and foragers are actually lacking fat storage. So this is why we can rule out the adipokinetic hormone. Then we know that insulin levels correlate with the foraging activities, but the insulin system in honeybees is very complex, so I won't speculate too much about how it might interact with SNPF. We also know that biogenic amines such as octopamine, tyramine, dopamine modulate the sucrose responsiveness. So we might need to do additional experiments to know whether SNPF or those biogenic amines act upstream or downstream within each other. And there's, so we also ruled out the bitter gustatory receptors, because in bees, there's no homology for gustatory receptor tuned to bitter taste. So like I said at the beginning of my of the results when I presented the different treatments, that we used sucrose solution spiked with a bitter compound, but actually here I'm saying that there's no bitter receptor in the honeybee, or there's no homology for gustatory receptor tuned to bitter taste. But some results in electrophysiology, like Gabriela did, showed that when you have a mixture of a bitter compound, such as salicine or quinine or caffeine, and mixed with a sugar, you have an inhibition of the gustatory receptor neurons activity tuned for sucrose or sugar. So to entangle this, we would need to do electrophysiology on gustatory receptor neurons upon SNPF treatments. And this is what I started to do a year ago in the summer, but I didn't have time to do all the groups. So I only, because I've learned that electrophysiology is super long. And it's really hard to do. So I only managed to do the pea-fed bees and the starved bees. And we actually stimulated the gustatory receptor neurons with increasing sucrose concentrations. And we recorded the number of spikes upon each stimulation. And as you can see, for the highest sucrose concentrations, we have a number of spikes that is significantly higher in starved bees than in fed bees. So we would need to continue this experiment and to do also the fed bees treated with the neuropeptide to know whether SNPF could actually act at the peripheral level. But it can also act at the central level, so in the subesophageal zone, which is the center for taste processing in insects. And this would require actually in vivo calcium imaging upon SNPF treatment. So now for the odor perception, we could wonder whether SNPF could also exert an action at the periphery, so on olfactory sensory neurons. For instance, in Drosophila, you have both the peptide and its receptor that are expressed in olfactory sensory neurons, with SNPF that facilitates the presynaptic transmissions in olfactory sensory neurons under starved conditions. And this actually leads to a more robust food search behavior. But we don't know whether SNPF and its receptor are expressed in olfactory sensory neurons in honeybees. So we would need, for instance, to do immunochemistry to localize their expression. We showed that the SNPF modulates the neural activity of the antennal lobe. But we don't know whether it acts at the level of higher order brain centers, like the lateral horn or the mushroom bodies. For instance, the lateral horn mediates the responses to appetitive odorants, but we don't know whether SNPF receptor is expressed in the antennal lobe nor in the lateral horn. And then we showed an effect of SNPF on visual memory formation of bees. And we could wonder whether SNPF could act at the periphery, such as the optic lobe, which is the first site of visual processing. So in the desert locusts, SNPF mRNA levels decrease upon starvation. So it works actually differently than in bees, for instance. So when the locust is starving, actually the level of SNPF is low, contrary to what we demonstrated in the honeybee. And there's a decrease. So there's a decrease of the SNPF mRNA level in the optic lobes under starvation. So again, it would be useful to do immunochemistry in the optic lobes of the honeybee to know whether SNPF-R is expressed. And the results that I explained during the visual experiments suggest that SNPF could act on the circuits that are engaged in the memory formation. And so we thought of the mushroom bodies because they are the center of associative learning in insects. So we can hypothesize that SNPF could interact with a molecular pathway leading to memory formation. So we thought of the phosphokinase A because it is known to mediate the association-dependent memory formation in foragers, but we don't know whether SNPF interact with this phosphokinase A. And we would also need, for instance, to identify, to know whether there's in the optic lobes neurons that mediate the reward, like we have in the subesophageal zone. So for the perspectives, we could do RNAi knockdown of the receptor SNPF in starved foragers, and our hypothesis is that they would behave as fed foragers. So if you inject RNAi that block SNPF receptor in starved bees, they would eat less, and they would have also a low sucrose responsiveness score and a low olfactory responsiveness. And I also wrote that we could do additional experiments for olfactory learning because I spent a few weeks and months to do olfactory learning upon SNPF treatment because we thought, okay, we have the olfactory responsiveness that is increased with SNPF treatments, we have also something going on. We have an increase of the projection neurons activity upon SNPF treatment when stimulated with odorants. So the next step was to see an effect in olfactory learning. But so far, the results I've got are only mitigated. So we would think to rethink the experiment. But overall, the learning performances of my bees, even the star bees, were not as good as what we expect usually. So we might need to redo this experiment with other odorants, or maybe to find a way to administer the treatment in a different way to have it directly in the brain and not in the whole hemolymph. So we need to think about a new way to perform this experiment. And so if you remember well, during the introduction, I said that it's interesting to study this neuropeptide in social insects because their behavior are mostly modulated by social cues. But here I only presented individual responses. So one of the perspectives could be to study the SNPF signaling at the colony level. For instance, does the diet quality affect the SNPF signaling? And do social pheromones release, for instance, modulate SNPF signaling? So that's all for me. My supervisors again, Gabriela and Martin, that are online, I guess, still, and also the collaborators that have been of a great help during my PhD, and you for your attention, and I'm happy to take your questions.
Yeah, thank you very much, Louise. I mean, that was a nice talk. Are there any questions? Okay, I know that was clapping your hands. Okay, Louise, then let me ask you a question. So you have set it up as that compared to Drosophila, that SNPF could have two opposing functions, so to speak. So one for affirmative behavior and the other for aversive behavior. And kind of now you kind of like the basis of your experiments, you don't find that in honeybees. So there could be two reasons for that, right? So one would be, okay, kind of like a different neuropeptide kind of like took over that function. But it could also be that that function is not there. I mean, so have you ever thought whether it could be a biologically meaningful finding, in the sense that kind of like honeybees respond to aversive stimuli differently than Drosophila, and maybe other kind of vertebrates, basically solitary animals?
Yes, so for instance, we could redo this experiment, but with the release of alarm pheromones, for instance, to see whether, because we know that when you deliver alarm pheromone, it has an effect on aversive responsiveness in honeybees. And maybe our experiment was not the most well designed to answer our question here with the effect of SNPF. Like maybe there's a counter effect of the alarm pheromone of what's happening in the bee when it receives a shock and the SNPF. So if we, for instance, if we have the same setup, but we add the alarm pheromone, so we increase this aversive signal that the bee is perceiving, I don't know if we could see an effect, but definitely the action of alarm pheromone would modulate the aversive responsiveness. So maybe we would see something of maybe SNPF could, I don't know, rescue the level of aversive responsiveness, or it could not act at all, and it could have no effect on aversive responsiveness in honeybees in general, because it's mostly regulated by social cues.
Okay, so
individual needs.
My question was meant a little bit differently. So whether, for example, foragers in honeybees, and maybe Martin knows. And
you mean that we should have tested it on
that they
on
are
guards? No?
that they are more resistant to aversive stimuli because they are collecting for the colony or something like that. So and whereas a solitary insect maybe has to be kind of like more sensitive in deciding whether kind of like to stay with a positive response and not responding to an aversive response. So whether there's kind of like a difference. So in that sense.
Okay. Yeah, that could be the case because I mean, I think we should maybe also try a bit more in aversive responsiveness, but with, I don't know, another kind of design, and maybe not, individual responses like we did, but with maybe at the colony level, or to see if there's an effect. Because the only thing we could compare was to solitary insect, but we don't have any other data on social insect in aversive responsiveness that are modulated by neuropeptides, for instance. So that could be that bees are not sensitive, that foragers are not sensitive to aversive stimuli, but we also know that biogenic amines like dopamine are modulating, aversive responsiveness and so on. So it might be that either we should do this at the colony level, or that SNPF has no effect.
Okay, good. So now there are other questions. So the first one is from Margarita Olova. Margarita, you can kind of unmute.
Hi. Wonderful talk and very interesting. The question that I wanted to ask is, from your results, it shows consistently that neuropeptide F makes normal bees behave like starved bees. So it pretty much turns a physiological not stressed bee into a physiological stressed bee. What are the, let's say, mechanistic drivers of starvation response in bees? Like, I don't know, how does a bee under normal conditions know she's starved? Is it like crop distension, stomach distension, midgut distension? I don't know what else. Is there any idea on how does the bee sense that she's hungry?
So there's few things. So we know that, for instance, insulin could be is a factor for honeybee starvation, because it's very because the signal is very strong when the bee is starving, but insulin is also very strong when the bee is feeding. So it makes the bee very sensitive towards any cue related to foraging activities. There's also signals from biogenic amines, because, for instance, you have octopamine that renders the honeybee more sensitive towards olfactory cues. And we think that it's also related to the crop volume, because obviously when the bee goes outside the hive, it has basically only the amount necessary that is necessary for the flight, the energy for the flight, because it needs to be a bit starving to be interesting for foraging activities. There's also cues that are sent by the larvae, like, okay, now it's time to go foraging. So what I mean is that there's it's not only the bees itself that is starving, because it responds to many cues. In a physiological point of view, so you have the bee that has the crop almost empty, and the low fat storage. Like, the nurse bees have fat storages, and when at the onset of foraging, they lose the fat storage, or it's a cause or consequence, I don't remember, but this is also a signal. So I don't know if I've answered your question, but there's many factors that are involved to make a bee starving.
Yeah, you did answer my question, and a follow-up question to that is whether there is differentiation between the bee's own physiological starvation and the signals of hunger from the colony, whether, for example, the signals from the larvae like methylolate, etylolate, all the stuff, does something different to the function of neuropeptide than the bee's
So that
own.
would be the next step, actually, because we haven't tried it. But that would be nice, to redo, for instance, the gustatory experiment. But at the same time, we sent the bee these kind of pheromones, like the larval pheromones, to see whether it increased, the response. We could do, for instance, the starved bees with the RNAi knockdown of the receptor, SNPFR, and then you inject, you blow this this pheromone and see which one has the upper hand. You see which one, yeah. But we don't know basically how this pheromone would interact with the neuropeptide.
Thank you.
Thank you.
Okay, so Christoph, did you have a question?
I can wait with mine.
Okay, so then Vidor.
Okay, so this is kind of, you could call it a suggestion. So like rather than concluding all, like rather than already going towards the hypothesis that perhaps the like aversive responses is entirely from social cues, I mean, maybe one could try seeing what dosing like guard bees with SNPF does to their aversive response.
So yeah, so if I understood well, we haven't tested the guard bees, but that could also be an option to see whether SNPF could act on those specific casts. But I would say that we would need to do something. We should maybe start to push the experiments towards a colony level rather than individual responses. So I would, if I had to redo an experiment with aversive responsiveness, I would start to use alarm pheromones and yeah, maybe guard bees to see whether they could respond more to this kind of signal with the SNPF interacting. So I don't know if I've answered your question.
Yeah, okay. I have got my answer. Yeah, thanks.
Thank you.
Okay, so there's a
I
question.
have a question in the chat. Sorry,
good.
Axel.
No, go ahead. Please.
So first of all, thanks for a great talk. Oh, this is by Marisol. First of all, thanks for a great talk. I'm very impressed for the great work you and collaborators have conducted. Second, I have a small question about the results of the experiments on the appetitive visual learning experiment. Have you considered previous studies in the literature on floral constancy in the honeybee, specifically that these insects may exhibit differential responses to blue and yellow depending on the color context or color scheme used to test the reward learning?
Yes, so actually, so when we trained the bees, so obviously we took foragers, so bees that have already an experience, that have been foraging outside, and we next trained them to a feeder, and then we put them in the feeder and we train them to go towards our Y maze. But when you see in the learning curves, actually, when you start at the first trial, there's no preference towards the blue or the yellow color. So if I answer the question, like our bees, when they started the conditioning, they had no strong preferences towards the yellow, for instance. So we made sure of that because first we used a different blue, and we had this big bias at the very beginning of the training phase. So we chose another blue, and then we we kind of get rid of this bias, but then we saw this bias in the memory tests.
Okay.
I don't know if I answered the question. But...
Yeah, there's no response at the moment, Omera.
If there's no other question, or Axel, do you want to go first?
I wanted to ask one thing about the visual learning experiment, which I might have missed. So, because you demonstrated it was a closed loop, that the bees are not going back to the hive, right? So, did you make them regurgitate the sugar water that they had intake, or?
No, so we gave them like only 1 microliter each trial.
Oh, okay.
So it's a very, so yeah, we have this kind of lack of motivation from our fed bees because they were already a bit fed. And then the reward was somehow, yeah, it was very, very small, but we couldn't really increase the amount of reward we would give to the bee, because then at the 5th trial, they would stop, you know, they would just stay there and do grooming or that kind of.
That was why I asked. Okay.
Yeah.
Okay, Christoph.
Yes. So very interesting talk. I think I learned a lot. I have a methodological question that has to do with your topical treatment. And I think you did this very interesting and important control to show that it is actually found in the hemolymph.
Yeah, with the chromatograph analysis. Yeah.
And I'm asking because we occasionally have the same questions when we, for example, feed bees with a certain treatment. Is there any data on how much of this is showing up in the brain? And also, and not just whether it's showing up, but I was wondering, is there maybe selective take up in certain parts of the brain? So that this could be, sorry, this is one part of the question. And my second question related to that is, do we know whether the uptake either in your study or in any study that has used the same method increases compounds in a realistic range, or is it kind of, are you creating a super quantity that could lead to an overestimation of the effect, or could it be the opposite, that actually you only manage to increase it a little bit so that what you find is an underestimation of the importance of the peptide?
So for the first question, where does it go, right? That was the first part of the question. So we don't know where in the brain it's going precisely. We don't know what is the amount that is going. We just know it's there. We know it spreads within the bee's body via the hemolymph. There's this paper from Andrew Barron, 2007, that he compared the topical application, the injection, and the food ingestion of biogenic amines, and that was the best method because it was the non-invasive one, and the drug reached the brain as well as, I mean, it reached well the brain. So yeah, we know it's going there, but we don't know which amount and what it reaches. And for the second part was about no, I forgot.
I guess it's related, but when we did similar experiments with, for example, dopamine and dopamine, you see a behavioral effect, but what you don't know is the quantity.
Yes.
Yeah, is it a realistic quantity that you're creating, or
I would say yes, because if we, for instance, when we look at the ingestion experiment, we gave the possibility of the bees, to drink up to 100 microliter, but it's an overestimation of the crop capacity of the bee, because it's like around 60 microliters. And with our treatment, some bees, I mean, the starved bees reached 60 microliters. And some of our bees treated with the neuropeptide also reach this amount of food consumed. So I would say that we have already 2 doses, one that is 10 times higher, and this is with this dose that we see, you know, an effect on olfactory, on imaging with the antennal lobe, with memory retrieval in the visual experiment. So I would say our doses are realistic. What we would need is something in between, like 5 microliters per 5 micrograms per microliter, to see whether sometimes, you know, we have this kind of asymmetric results, to see whether sometimes it could be just in between. Yeah.
Thank you.
Christoph, can I make a comment to that? So I remember that I once had the question kind of like, so if kind of a neuromodulator kind of has diverse function, right? And kind of like you do your behavioral experiment, but then kind of like, I would say, as in Louise experiments, then you see an effect on feeding, and maybe it's not showing an aversive response. How is that coming, right? As if the neuromodulator could initiate different behaviors. And I don't know with whom I discussed it, probably, but more knowledgeable than me. And this person said that there could be something like that, you know, injected or applicated neuromodulators will kind of like have a stronger effect on neural circuits that are active. Yeah, so, and if you have kind of like, you have this behavioral context, so kind of like the neural circuits involved in that behavior are more active, and they might take up more of that, so that you have then kind of like an increase of that behavior, and not one of a kind of like a different behavior in which the neuromodulator also plays a role. The neural circuit is off at that moment, right? And so that might be a reason why kind of like, how to say, you get either this yes or no response, and kind of and not a totally different behavioral response. I mean, I don't know whether it's true, but it sounds kind of like a little bit plausible.
I guess it's one of the problems that we have, and this is not a criticism of your study, because we do exactly the same, that it is still, in a way, a crude method. And
yeah, it would be more easy. It would be easier if we had like genetic tools like in the Drosophila, you know, but in the bees, it's still complicated. So we do with what we have, and we try to be like less invasive, and well, no, it's not as we are never sure about how the drug is acting or where it goes, because we can't really control it. Right.
Yeah, it would also be great if you could just inject it in the tissue that you're interested in, but I guess that's also very, very difficult. Yeah,
because even when we even in calcium imaging, like when you inject somewhere, it spreads, so it's quite difficult to be super specific.
I think how to say, so this whole question of neuromodulation is very interesting. And I mean, your lab, Martin, is still there, and a nice dopaminergic paper. Congratulations, by the way. But I think that so it's interesting. It's a really interesting question, where to go from here, right? If we don't have the tools that are there in Drosophila, and right, kind of like everyone writes it. Okay, if we have kind of like CRISPR-Cas9, then we can do it. But how to say, this is kind of an easy sentence to write. But the question is whether one is really taking up that, or you know, or kind of testing the function in all behaviors. So I think it's really the question where to go. So I liked kind of like your schematic, kind of taking from the Drosophila, putting on to the honeybee, right? Where you have a kind of like hypothetical pathway. And so then I would say, okay, it would be a nice question whether, for example, the octopaminergic neurons, so these VAM neurons,
The von
I expression the SNPF receptor.
Mm.
Yeah. Or when kind of like, okay, we don't know what are the dopaminergic neurons that came up in that. So seeing whether we can get some of the dopaminergic neurons and kind of seeing what kind of receptors they have.
Yeah, for instance, in the Drosophila, you have dopaminergic neurons that go to the lateral horn, and they express NPF, and they modulate the responses to others that have, you know, an intrinsic appetitive value for the Drosophila. So it's super frustrating when you do all the literature, because there's plenty of research in Drosophila, but you can't really transpose it to honeybees. I mean, you can a bit, you can get the inspiration and get ideas and speculate and discuss about this, but you can't really say, okay, it works the same in honeybees, because, I mean, it might, but we don't know that. For instance, we don't even know in honeybees how NPF works. Like there's no receptor, so does it also bind to SNPF receptor? We don't know. So yeah, it's like, I mean, we opened large discussions with this topic.
Yeah, and the problem is that kind of like no single lab can do all that, right? So, and then the community is small, kind of compared to those.
Martin wants to talk.
Yeah, okay, good. Let's hear Martin.
Thanks a lot, guys. First of all, thanks for organizing this. This is a great, absolutely great series, actually, and great initiative. And you two are doing a fantastic job. I'd just like to comment on what you said, Axel. So, which is, where do we go? I mean, this is an essential question. And essentially, I mean, especially if you say, as you said, where do we go if we do not have the tools that Drosophila has? And that's a fundamental question, because actually we know that we depend on grants, on funding, and so on. And it's becoming difficult in the light of this success. And we are happy for our Drosophila colleagues, but of course, it makes also life a little bit difficult somehow. I think that a place where to go is to insist in the components that you might not find in Drosophila, even in the absence of tools. What I mean by that is that we have here a model which has an essential social component, which has an essential social regulation, central place foraging, navigation capabilities, communication strategies, and all these elements are totally absent in Drosophila. So I mean by that we have a rich environment or landscape to explore, being somehow independent on Drosophila. So if we ask questions about neuromodulation, I think the strategy should be to place them into one of these contexts where we would not compete with these guys. So you raise a very interesting question, actually, which is, I mean, when you say, okay, do you think that, for instance, the absence of modulation for aversive stimuli might not be there? And that's a very interesting question, actually, because actually it opens the possibility to think about the control of aversive responsiveness by social pheromones, and not necessarily on an individual basis, but regulated socially. And that opens a fantastic possibility and a new world, actually, for asking questions. So I think this is where we should go with these neuromodulation questions, actually, to place them in this unique universe, to say in this way, and try to do the best we can within that universe. I don't know if I'm clear enough. That's the way.
I agree with that totally. You know, I mean, how to say, the behavior is way richer, right? There's no question. And particularly, it's complicated behavioral, I mean, showing complicated behavioral capabilities in a behavioral experiment, way easier in honeybees and in Drosophila. Almost you cannot do that, right? So I agree that, and if you say that the social component and getting it, putting it into a different framework, that might be a way. Yeah. And so here's something that I actually learned here. So that kind of like, here's no, I mean, only a few insect people, and then these are all vertebrate and then kind of molecular biologists. And when I once gave a talk about honeybee foraging, right? And then I got the question from someone, don't they get fatigued? Don't they get tired and stop? Yeah, so that made me kind of like actually aware of what kind of specific behavioral phenotype foragers are. Yeah, and I think that so in the B groups, kind of we take foragers as foragers, right? But that the forager are an extreme behavioral phenotype, right? Constantly kind of like highly motivated, which is actually good for kind of the learning experiments, right? But you hardly see it in any other animal, right? Except maybe other social insects. So it's also something to think about.
Absolutely.
Okay, yeah. Christoph?
Any other questions? Well, if not, then I think I stop the recording.